So then we are back to the last presentation this morning. Back to Kristian Hveem. And this time it's about sequencing, storage, and access to large data sets. And you have already been introduced, so please. Lunch is at 11:30. So we will have time for some questions, I get, towards the end of this presentation.
Yeah, absolutely. Well, thank you. I'm actually quite proud of my first picture. But I stole it from Ole, not the picture, but the background from Ole Petter Ottesen. We had a breakfast meeting at Arendalsuka, and he had a lot of those moving backgrounds. So. I'd just like to give you a brief outline of what I will cover, and I hope that will basically do that in this order. I will start with the challenge. I will talk quite a lot of transformation of data or to data from biological samples, what you could call digitizing biobanks. Then some comments to the best strategies for genetic analysis. This will not at all be exhaustive, just some input and share some thoughts. Then about sharing of data, some of the legal aspects as well. Building up comprehensive data bank resources for reuse, second-hand data, both phenotype and genotype data, safe storage of sensitive data, establish the best access and data processing procedures, keeping data close to the donor community, and potential of collaboration. To me, this looks like topics for a whole seminar. So, there is a challenge. This was published in Nature last year, and I think it's very fascinating. This is a table. What it illustrates are the numbers needed to treat to have one person that has full effect of medication. And if you look at the medications that are listed here, for instance, Nexium is a proton pump inhibitors. You can see there are 25 people that sort of sacrifices themselves for the effect of this one. This means over time, if you look at statins, only 5%, only 1 out of 20 has effect. And you can see it's the same picture for many of the more regularly used medications, and not to mention the anti-TNF alpha or the biological treatments. They're also very expensive, and they tend to be, patients tend to use them longer than they are supposed to, because the doctor feels sorry for the patient if the patient doesn't have effect of the medication. So, well, we can try a little bit more and a little bit more, but it costs a lot. And basically, what the patient will experience are side effects and not effects. So this is not good. And if you look at statin treatment over 5 years, this is primary statin treatment before an event. I think the picture is different after myocardial infarctions, for instance. So 95% have no effect. It's terrible. In 1.2%, one life was saved. In 2.6%, one non-fetal heart attack was prevented. So 97.4% took the medication for this one non-fatal heart attack. In 0.8%, one stroke was prevented. 1% developed diabetes that would not have diabetes. 10% developed rhabdomyolysis, which is a pretty serious condition with disturbances of muscular tissue. And, of course, at significant costs. So I'm saying all this because I think we have part of the solution. If we can study genetic variation amongst population-based study participants and link this to these different disease endpoints and phenotypes, we might be able to predict upfront who shall be treated and who shall not be treated, and who will have the more serious side effects and who will not have the more serious side effects. So we should develop a more precise understanding of how genetic variation contributes to disease development amongst some and not amongst others. This is how I would define precision medicine. There are different ways of talking about precision medicine, personalized medicine, but in this strict definition, we talk about how genetic variation plays a role as sort of the bottom of this, all these layers. Then we would also like to stimulate a robust scientific understanding of health and disease by prioritizing genes for functional follow-up. To identify genes with functional effects by an bioinformatics approach, by Mendelian randomization, or through mouse models. So we are getting there. We have used these tools, we are using these tools, and we have also been funded by the Biobank program together with Magne Thoresens group on applied biostatistics. And I think by doing this and by joining forces on the national level, we might have a chance to add our knowledge and to compete with other groups internationally. And related to the previous picture, we'd like to identify new drug targets based on genetic analysis and also have a look at known drug targets to see the genetic variation related to that. So if you go back and or memorize what I said in the previous presentation, that we have genotype 170,000 Norwegians. Some of them are from quite interesting populations. They could be what we call populations with a bottleneck effect, meaning that out of a parent population there would be some genetic drift. There could be a drastic reduction in the population. Some could move out, some will stay put, and surviving individuals will have a different genetic development. And we can also talk about isolated population, as this poor fellow here, where populations also tend to develop different genetic constitutions based on that they lack the normal sort of input from other parts of the population, from other genetic pools. And we have always said that the Finns are a bit special. Are there any Finns there? And I think now they're looking at this as a great advantage. Because if you look at the Finnish populations and the non-Finnish European population in Finland, this is what's in this table. You see that there are significant higher number of so-called non-coding mutations or loss of function mutations among Finns compared to non-Finns European. Why is that a good thing? Well, it's a good thing right now because among these non-coding mutations or loss of function mutations, we find a lot of interesting experiments performed by nature. Genetic constitution has been altered in some persons, giving them not only a negative effect but also positive effects. So I've previously talked about a protein PCSK9 where there is an inhibition of the production of this protein, which has a based on this loss of function mutation, and this has a very good effect on lipid levels, cholesterol levels, and is also protective against cardiovascular disease. So nature can do some very interesting experiments in some persons. By doing this large-scale genotyping, we're able to identify a number of these experiments and see how they can be applied in better insight and understanding of different disease mechanisms. So that's why it now counts to be a bit special. So now we're revealing the hunt data. We're doing a primary analysis on these 70,000. We have genotyped around 20 million markers against all these phenotypes. So we hope that the people in Nord-Trøndelag also are a bit special. So we can find some very interesting hits. We don't know yet. So what would we recommend? What is the best strategy? Should we continue to genotype? By genotype, we now mean typing a few 100,000 markers along the genome and exome, compared to reading all the different genetic markers along the entire genome, as we would do if we do whole genome sequencing or whole exome sequencing. And I think Iceland is a good example where they have now genotyped more than 100,000, and they started sequencing around 25, maybe 30,000 so far. And I think now they're basically stopped sequencing Icelandic samples because it doesn't pay off. At this stage, it's probably not cost effective to sequence even more. It's probably better to have a bigger population, which they don't have. That's why they're so interested in the Norwegian populations and other Nordic populations. And for us, to genotype a lot more than we have genotyped already. And do sequencing in parallel, but I think genotyping should be the priority number one in terms of what sort of genetic exercise or genetic approach should we do in the future. I'm primarily talking about population-based studies, but this can also be done in clinical biobanks, of course. I talked to Dag the other day to just see how much is actually done, and it's very little that's done in Norway in terms of whole genome sequencing, a little bit more whole exome sequencing, but I think these are pretty representative numbers. These are from blood. I'm not then including these tumor sequencing project with Ola Myklebust and others. But there is an increase, and as Dag said that in 2015 you guys did 400 whole exome sequencing. Now you've done 700 so far already in this year. So there will be more. The instruments are improving, and the costs are dropping. And then there's an increasing number of ongoing extensive genotyping initiatives and functional follow-up based on high-throughput proteomic and metabolomic platforms. And I think that's an area, or those are areas where we should put a lot more strength and efforts from now. And that's why it's really a great pleasure to listen to these presentations from the Janus Biobank and to see how technical and analytic progress and development has been so much in favor of Janus Biobank. Because a few years ago, you were pretty much thinking this has no future. We don't have DNA, so. And now we're even doing genotyping on these serum samples. So these omics, high-throughput omics platforms are very favorable. They do a lot, they analyze a lot, and they use very little material. But this creates a lot of data. So we need to have a good strategy for handling data, storing data, sharing data. And I would also say that there is, and by right, a skepticism on how to share and store genetic data. I mean, we have to be very considerate. But proteomic data and metabolomic data will be just as sort of risky in terms of protecting sensitive information. So far, as far as I know, two major establishments concerning safe storage of sensitive data. The first one out was TSD, Services for Storage Sensitive Data at the University of Oslo. and where Gard Thomassen has been responsible for that, or a bit for that establishment over several years now. I think this is a great achievement, and it is a very useful tool that many of you are using already. It's not tailor-made for genetic studies. It's a more genetic solution for all sorts of data. So at Hunt, we have been making a little bit more specific solution for what we call a Hunt data center and a Hunt computer cloud for our increasing numbers of data from all these genetic studies. And so far we have also been storing samples from or data from the Harvest project. This is now being transformed over to TSD, at least some of it. I will not go into any details here, but I will just emphasize that there is a strong focus on this also in Norway. I think we're doing pretty good. And I think politically this is a good signal saying that we would like to build solutions close to where the data are generated and produced. This is something I mentioned in the beginning of my talk, to be able to keep the data close to the donor community and not sending them all over the place. I think this also has some impact on the recruitment of future studies. I'm not saying that local storage in all aspects is better than remote storage, but I think it means something for the people involved in the studies. And then at Hunt we have been building a data bank for all the other data sources, the non-genetic data. And this is now a very comprehensive data bank where all the details about what's stored, the representativity of the data from all different Hunt studies can be looked up on the web. And I think the most important message here is that this is a very nice resource for others that have not primarily done the analysis, but can do a lot with the data in different aspects and related to different research questions. And if you look at the mother and child study, this is not in place. They would certainly like to have it in place, but so far it's not entirely in place that data generated from the mother and child study should be brought back to the National Institute of Public Health and stored there. Eventually, I think the IRB will accept that the data is brought back, but from in Hunt, this is a requirement to all the different projects PI that they have to bring the data back. And to make sure we get the data back after the primary analysis, we don't give them all the other variables before we have the data stored. We don't use it, but then we have it. So there will be an embargo, but it's a pretty good way of getting data back. So as I also mentioned, digitizing biobank by converting biospecimens to data is happening in full scale. It is, I think it's a nice development. It's easier to handle data to some extent than samples in freezers. It's somewhat cheaper, even though storage costs will increase also dramatically. And we have to consider that when we apply for funding now in the future, that we need infrastructure funding for storing data. Arvid Hallén mentioned Denmark and Sweden, but Finland is really doing well in bioinformatics and data storage, data handling. I think it's even still paid for by the Minister of Science, which I think is a very attractive feature, because these costs, as I say, will increase. Now this is perhaps a bit too complicated slide, 15 minutes before lunch, But what I'm trying to, or what we are trying to illustrate in this is different ways of storing and sharing data. So if you look at the right-hand side, this is what I've been talking about now, TSD, Hunt Cloud, Mostler in Sweden, Aputa in Finland. These are secure computer systems where data are kept on owner servers, offering secure processing capacity to external users. Then on the left-hand side, we have a suggestion of presenting summary data in a result server, where we could present pre-analyzed data from big data sets and look at all genetic data against entire registries. These will, of course, be encrypted and filtered and so forth, but this could be something that could then be publicly available. And then there is this one. It's an analysis server where we can do interactive analysis of data for predefined analysis. Individual data will be kept behind firewall. And I forgot to translate that one. But this could be a solution for data that are too big to be moved, so you can create different systems for doing these analysis. I fully understand if this now turns a bit complicated, but I think what is probably most interesting, also in relation to what Arvid Hallén talked about, is this one, the Cohort Explorer. And this is similar to the Danish Biobank registry. I just think I saw Mats Melby enter the room, yes? Welcome. Where a researcher will like to make a request, make a data query. One way of structuring this is that the data could be then deposited into a sort of a neutral room, and then the data owners, they would frequently ask into this room, do I have any requests of today? Anything people would like to know about my information about my available data? So the idea is that if you would like to do some research, I have one concrete example. I've been asked to provide data into a pan-European cancer project. We're looking at gallbladder cancers. I think we have 7 or 8 in Hunt, and there are a number of cohorts. I will obviously need to join forces to increase this number to a significant level. Now I asked the IRB for permission to ask the cancer registry whether we have 5, 6, 7, 8 cases. It would have been, I think, very useful if I just could use this model, make this request, and within a few minutes I will get the answer back. There are 5, there are 2, there are 3. So I can then see whether it is worthwhile to file a full application and get real data. And we have discussed this with the data inspectorate, and they are very much in favor of this because they feel that will probably stop some of the projects by filing a full application and getting real data, because you can immediately see that the way you are asking this research question and the phenotypes you would like to do your research on, they are not in sufficient numbers. A part of this activity has been funded by the infrastructure project, so we're also working on this now to build such a solution. And I think also E-helsedirektoratet are doing something similar. So in December we hope to meet up with the E-helsedirektoratet to see if we can come up with a common solution. And I was really excited when Helge Hveem at the Data Inspectorate said that he even helped us with technical solutions there. So I think a lot more than I had expected, actually. Then something around legislation and ethics. It says Heath Research, it's Health Research Act, the Biobank Act, that applies for clinical biobanks, and the Biotechnology Act are all acts that we have to relate to, and not to mention IRB, so REC, that they relate to these different projects to try to find out which one is the correct one to use. And when you do genetic studies, large-scale genetic studies, population-based, you end up somewhere in between, because you might identify some genes that could have predictive effect on some of the participants. One could be familiar hypercholesterolemia, where this has also been mentioned previously in a meeting here at Litteraturhuset, where IRBs or REC said that they were not sure whether we were allowed to do that, because they thought this might be something that should be governed by the Biotechnology Act and not the Health Research Act. And the difference there is that if the project belongs under the Biotechnology Act, you shall then pre-inform the participants about the fact that you now are doing some genetic analysis and might have some effect, and you should ideally have a response up front whether they would like to have that information or not. And then, of course, you need to inform them. And that should be performed by a specialist in medical genetics. We don't have too many of those in Norway. So this is a challenge. And we also have to do quality check on the data and on the analysis, so we are absolutely sure that we are informing the right persons, that we have not switched tubes in the biobank. So this was sent to the Health Directorate by IRB in Central Norway. And instead of saying this is the act you should use and not that one, so they responded saying you should consider this more ethical and not juridisk. And I like their response, because I think that's exactly the situation. And then you come into this term actionable findings, which has been nicely described by the new guidelines from NEM that came this spring. Also putting more emphasis on the facts, or how this might affect the participant, and not to that same extent be too occupied with what different act that would apply in this specific case. And NEM also stated that genetic data is basically not anonymous. And I think that's a wise decision. We agree with that. That means that even though it's de-identified and anonymized, we know that there are ways of getting around it and probably do some backwards identification. So the emphasis should be on data storage and data safety. And we have also argued that when we find these genetic markers that might have some predictive effect in a population-based setting, we don't really know, because this has not been tested out entirely so far. We know that only 1/3 of those with familiar hypercholesterolemia has been identified in the Hunt cohort. So there are about 80 that are known. There are probably 160 that we have no knowledge of. And that is probably not because they're all dead. That might be because there is a different genetic penetrance, and the clinical effect of this, of having this, being a carrier of this gene, is not straightforward. So this is also our argument towards the IRBs, that we would like to do a little bit more research up front before we consider reporting back to the participants. So data sharing is also a challenge. It's a challenge for the participants, it's a challenge for us, and it's a challenge for IRB, especially if you share data in databases as dbGaP, because then we are not informed up front of who have asked for access to data, which is a sort of an unbroken principle in Norwegian research legislation. But we have asked the participants whether they would comply with this or whether they would mind sharing data in such a database, and most of them had no objections, meaning 15 out of 95,000 had a problem with that. But I think that instead of moving more data to these databases, we should probably try to construct a solution where we keep the data on our servers, but we can then give other researchers access to the data where we have full control of them. Then there is EU Data Protection Act. You've probably heard about that one. There's been a very intense and pretty exciting discussion over several years now. We think that this will turn out acceptable, that research will still be exempted, and we can carry on what we've been doing for many years. Then Data Inspectorate and others, ourselves included, want to put more emphasis on donor communication. So at this latest infrastructure call, the Biobank community sent an application to the Research Council to build a MyPage solution together with the eHealth Directorate for all biobanks, all research participants within clinical biobanks and population-based. This is now coordinated by Tromsø, and we hope that we can get at least some funding, which will put us in a very good position to make a better solution for donor or participant communication than we have in place today. And in fact, the Data Inspectorate, they have told the Hunt4 project group that they will not approve of a Hunt4 study before this MyPage solution is in place, which is a good incitament to get it working. Another ethic challenge is how to handle data within clinical biobanks, where most of the samples available are not consent-based. And this brings me back to my first bullet point, the Biobank Act, because the Finns did something remarkable in 2013. They changed the rules for using data from clinical biobanks, introducing an opt-out policy, saying that data deposited there would be available for research if the patients had not exempted or opted out. This is the same principle as they have in Denmark. And from one day to another, the clinical biobanks were sort of moving from far behind till be in the front of a lot of exciting research that's going on in Finland. We're meeting this expert group by the health ministry in a couple of weeks, and I think this is at least something this group should look into, whether there is something we can do on the Norwegian side as well to make this easier. Okay, I'm just about to finish. Access. A few words about access. I mentioned access during my presentation of the harvest data. Access should be easy. We talked a lot about that. Arvid Alén has talked about a lot about that. It should be fair. That means that people should have access if they have a good research project. And the industry should also be granted access if they have a good research project. This is, I think, important. And we have to open up or open up for transnational access to collaborate across. And there are some hurdles there, some hurdles moving data, some hurdles in moving samples. This has to do with the the Statistics Act and other regulations that I hope that also now will be looked into in this huge national process for improving access. Networking is important. There's a great potential in Nordic biobanks. And if you look at this map, you can see that there are a number of samples that have been genotyped in the same way in different Nordic countries. So we're talking here about maybe 700,000 samples that could potentially be brought into one study. There's a variable number of whole exome and whole genome sequencing studies. Numbers are not that high. In Denmark, SSI and Mats Melby are planning for genotyping 1 million Danes. That will bring them in the front, by no doubt. But if we add all these together, we are already on a close to a 1 million level on a Nordic basis. So there are huge potentials. I think I'll drop that one. There is also a lot going on the European level, infrastructure development. And in Biobank Norway 2, which was funded last year, we have included Elixir and the health research project, as well as the National Sequencing Consortium or platform into several other work packages to strengthen this bond between biobanks and IT infrastructures and analytic capacity and data storage and IT management and so forth. So, in summary, there are a number of very promising precision medicine initiatives in the Nordic countries, and certainly also in Norway. We are still far from utilizing its full potential, but we are not at all where we were a few years ago, where Kari Stefansson said that in Norway we would be famous for having the best collections that were never used for anything. At least we're not there now. Biobanks are being transformed at full speed from samples to data, big data, and we're just at the beginning of having adequate solutions for data storage and processing in place. Thank you.
So Kristian, here's one more for you. Thank you very much for this very comprehensive and nice presentation. It's really important issues. I was sitting there wondering, what is the most burning issue here? What would you, say, we have to deal with this now? Is there one thing, or do we have to need the lunch to discuss that?
On top of my wishing list will be to have the ICD codes within 1 week and not in 6 months. Okay. So when we want to have access to all the different register data now, it takes such a long time. And there are access committees all over the place, so they want to have a saying, and that's fine, but it takes weeks. And then eventually the IT personnel in the hospital, called HEMIT or whatever, they are then doing the finalizing this job, and then they have to do something that's more important in the clinics. I can understand that too, but we have offered to pay for special personnel that are only ours within the system. I think that might work.
Okay, so that really draws things back to Arvid Hallén's presentation this morning.